Hello, everybody. My name is Verena Germann. We're here for the poster flash of the WetPol 2021, and I'm happy to chair you through this session. We will have about 3 minutes presentation of each presenter, and then we will collect the questions in the chat and address them in the end. We will have the first presentation by Angela Sofia Gomez Lopez, and please, Angela, share your screen and start the presentation.
Okay, let me a moment, please. I'm going to share my screen now. My presentation. Could you see my presentation?
Yes, we can see it.
Okay, I'm going to start now. Good afternoon to all attenders. My name is Angela Sofia Gomez, researcher at the University of Cauca. So in this important meeting, I'm going to present my research that is titled, as you can see in the picture, evaluation of the removal efficiency of a constructed wetland to purify wastewater from the wet mill of coffee in Cauca, Colombia. First of all, it's important to mention that the wet processing of coffee is developed in some Latin American countries as an alternative to obtain a high quality coffee. As you can see, this is the wastewater from the wet mill coffee produced in the Pomoroso coffee farm. However, this important process generates a variety of environmental issues, such as anoxia, algal bloom, of environmental loss. So in this coffee farm located in the department of Cauca, was installed as a secondary treatment system, a subsurface flow constructed wetland, such as secondary treatment system. The treatment tank was conformed by grass trap, after that an acidogenic lagoon, and after that we installed as a secondary constructed wetland. Okay, in this research, we already the removal efficiency of this system. The main characteristic of this system is that it was seeded with the species Phragmites australis. It has an area of 6 square meters, and it has an input load of 631 grams per day. So in this research, we evaluated the removal efficiency in terms of chemical oxygen demand, nitrates, phosphates, pH, and total suspended solids. Our experiment was of repeated measures because the samples were taken at the entrance and output of the system. After that, we transported our samples at the CRC laboratory. In this place, we analyzed our samples. Subsequently, we applied with COX some non-parametric hypothesis test in order to establish if our system complied with the national and international regulations, in this case, the Colombian normative of Colombian rules, that is Resolution 0631 of 2015, and of course that was given by World Health Organization. And finally, it was applied basic exploratory and descriptive statistical analysis in order to obtain the different graphics, statistical graphics, such as box graph, line diagram, and a couple of histograms for establishing or analyzing the behavior of each of parameters that we have evaluated. Okay, finally, according to our results, the wetland increased the pH by 2 units, behaving like a Bucher system. In terms of removal percentages, the chemical oxygen demand was removed by 82%, nitrates by 69.5%, phosphates by 79%, and finally, total suspended solids by 70%. So these percentages were in agreement with other investigations carried out around the world. On the other hand, when the Wilcoxon test was applied, the output distributions, it could be corroborated that our system was completely stable by removing pH and total suspended solids, but was a few unstable and a few inefficient in order to remove chemical oxygen demand and nitrates and phosphates, due to the fact that, you know, the wastewater has a particular characteristic and is that completely acidic. So this phenomenon is completely important to the removal of these parameters. So in order to conclude, our system removes suspended matter in an efficient way, of course, a chemical oxygen demand removal of was carried out mainly by anaerobic degradation, because the characteristic of the system, anaerobic system, and optimal phosphate removal showed that the gravel still had capacity of absorption, and the nitrate removal efficiency was influenced mainly by acidic conditions. And of course, that maybe in the future, it will be necessary to increase the inlet pH in order to increase the stability of our system. And thanks so much for your time and your attention. It's all my presentation. Thank you.
Thank you very much for your presentation. We will gonna go on with the next presentation by Anh Bui.
Okay, let me prepare to share my screen really quick. Okay, where is it? Okay, and then for this, I've actually have a poster to present. I'm trying to see how to put it on full screen. Sorry. Okay, I was able to do this earlier, but okay.
Maybe if you tried with the view or like that.
Sorry?
Yeah.
Oh, that didn't work. Okay, I'm sorry, that it worked earlier.
Don't worry.
Okay, there we go. Okay. Okay, I suppose everyone can see that now. So my name is Anh Bui, yep, and I'm doing my research on behalf of the Center of Environmental Research at Helmholtz Institute in Leipzig, Germany. And my project is called the ULVAT project, so accessible gray water treatment solutions in informal urban settlements using in South Africa using constructed wetlands. That's what my project is focused on. And my study focuses on the examination using horizontal subsurface constructed wetlands planted with Zanthedesia and Cypress and filled with rubble and macadamia nutshells for the treatment of gray water generated by informal urban settlements. And I'll go on to what that is. So some things are important to know and why this project was generated in the first place is that over 12 million people lack access to clean water supplies because in South Africa, they live in slums, which are the informal urban settlements. Therefore, there are minimal disposal points. The large population that generates an abundant amount of gray water and black water mixtures increases to the poor disposal of the gray water. And additionally, that also poses an environmental threat. So with that being said, with the larger populations, our aim was to examine how our 1 meter lab scale constructed wetlands are able to, are able to treat and handle these high gray water loads and their response to it, their ability to understand the flow behavior and how the substrate materials handle it. So important thing to know is our substrate materials consist of rubble from demolished buildings and macadamia nutshells. And these were materials that were collected actually in South Africa, high waste materials and autoclaved before being transported to our lab in Leipzig for examination. Also, the 2 plant species that were selected for substrate include Zanthedesia, which are native to South Africa, in addition to Cyprus. The Zanthedesia in particular, there's limited knowledge on that because they haven't been used for wetlands, but they serve as an ideal co-product for the residents who live there. So if these high loads in particular, so at the beginning of the experiment from the early stages after the preparation, assimilation, and adaptation of the wetland, we have seen initially an increase within the inflow to the outflow of the wetland by about 50% within a 3-month period. So that in turn shows that the system wasn't able to handle the normal loads generated by the population earlier. And another important thing to know is to understand our system, we did the examination among the sampling points, which include between the two plant species and from the inflow to the outflow along the flume at a constant depth. Later, we identified how the wetland adjusts to and readapt to after the high loading of the wetlands by incorporating a 4-month period of just loading the wetland with tap water to see if it would readjust to the conditions that we found were the oxygen recovery did not occur. However, the plants have stabilized in the system. While we don't have any explicit measurements of that, we have noticed over time with the higher loading, so the 100% overload and the 25% overload from point one, the point between the Zanthedesia and the points between the two Cyperus have decreased constantly over time with the loading and have stabilized. However, within the research, there needs to be more understanding within the bacterial species among the substrate and how they contribute to the degradation of the macronutrients involved. Thank you.
Thanks a lot. Thank you, Anne. We will continue with the next presentation by Annegret Budach.
Hello. I'm going to share my screen. I hope everyone can see it.
Looks very good.
Okay, good afternoon. I welcome you to my short presentation on the topic effect of hydraulic conditions in PFR reactors using bio-electrochemical processes to influence the formation of greenhouse gas emission. The content of the poster has been developed in collaboration with the EU-China project Electra. Since more than 2 decades, climate change and global warming are major topics in the scientific and political community. In order to exceed the goal of less than 2 degrees warming increase, it is necessary to stabilize and to also decrease the concentration level of greenhouse gases from 500 to 450 ppm in the next years. This requires especially the reduction of methane and NOx emission in all sectors, because they contribute most to the atmospheric warming, shown in the graph of the left side. So also in the wastewater treatment sector, we should not only focus on the removal abilities in our systems, but also take a look on the environmental friendliness of our systems instead. In our experiment, we used continuous inflow planted fixed bed reactors filled with conductive materials as biochar and graphite coke from Medfi company and gravel as a control to determine the methane flux under optimal hydraulic conditions. The idea was to use the enhanced electron transfer due to the material properties and increased anaerobic respiration processes to reduce the formation of methane in the system. In the experiment, samples from the gas, liquid, and solid phase of each reactor were taken. The microbial community was analyzed with the focus on methane producers, and it can be seen that the highest percentage of the methanogens were found in the gravel reactor with 81% in the liquid samples. The amount of methanogens from graphite coke and biochar were significantly lower with 23%. In general, the bacteria and archaea amount was higher in the liquid samples in our system. If we now take a look at the gas analysis, we can see that the highest amount of CO2 measured were in the biochar reactor, but the highest amount of methane, the graph on the right side, were not found in the gravel reactor as we expected due to the high amount of methanogens, but in the graphite coke reactor. That leads us to the conclusion that the snorkel effect in the system, so the flow of electrons, could not only go in one direction in the system, but increase due to the cathodic insufficiency in the graphite coke reactor. So it also goes in the opposite direction. Furthermore, the electrical ability of the two materials, biochar and graphite coke, are quite different. So biochar is more acting as a capacitor due to the chemical structure, while graphite coke only acts as a conductor in our field. And could increase the electron pressure in the anoxic areas of the system. That, furthermore, could lead to more methane formation in our system. I thank you so much for your attention, and if you have further questions for me, then please send me a message or take a look at my poster.
Thanks a lot, Annegret. And yes, that's true. So as she also mentioned, please feel free to check out the posters and the videos. There's some additional information for each of the posters, and most importantly, also to vote on your favorite poster. We will go on now with the next presenter, with Aday Amirbekov. I hope I said it right.
Yeah, thank you. Can you hear me?
Yes, we can hear you.
Yeah. So let me share my screen. Okay, this one should be right. Yeah. Can you see my presentation?
Yes, very nice. Looks good.
Okay. So my name is Aday Amirbekov. I'm a PhD student of Technical University of Liberec, and my work focused on HCH removal in biochar-amended artificial wetland beds. So the main objectives of my study was to compare to removal of gamma HCH and its isomers in different constructed wetlands at the heavily contaminated site, and also observe the development of microbial communities, tested setups, and monitoring of genes, which is encode enzymes, which is involved in biodegradation of these HCH isomers. HCH is a hexachlorocyclohexane. It's an insecticide, and it's a persistent and toxic compound. Our site, named Hájek, is located in region of Karlovy Vary in Czech Republic, and it has been contaminated in the 1960s, mainly by ballast isomers and some metabolites of HCH. So our wetlands was divided for two sections. Control wetland bed, which was filled with natural shallow water with soil clay substrate, which is obtained directly near the polluted site. And the second one was filled with biochar. So we used chemical analysis and DNA extraction to determine total bacterial biomass, and NGS sequencing to estimate the bacterial diversity. Results showed that biochar wetland bed demonstrate the high efficiency of the removal, 96% comparing to control wetland bed, which is 68%, and the efficiency even was increased to 98% when the initial high flow rate was decreased. This is chemical results. Functional genes. Overall, the genes which is encode enzymes in HCH biodegradation were detected in all sediment sample and water samples, and in most of Gallionella and sulfuricurvum. Also, the sediment samples were rich in Simplicera, Rhodoluna, and Rhodophyrax species. The chemical analysis of biochar showed the absence of any adsorbed HCH-related compounds. It can be therefore assumed that there were microorganisms able to degrade these compounds, and analysis of the sediment samples taken at the end of the experiment showed confirmed the presence of functional genes and bacteria genera capable to degrading HCH isomers and their byproducts. So the main advantage of the biochar wetland bed is in simplicity and effectivity, and also the speed of installation and the price. So this may encourage the use of these biochar wetland beds, for example, for temporary installation for water purification during the construction of other wastewater treatment technologies. Thank you. You can find more detailed presentation on my profile. You can find my poster as well there. Thanks for the attention. I'm finished.
Thank you very much for your really nicely illustrated presentation. The next presentation will be by Jan Nemecek.
Let me. How to do this? Sorry.
There should be a button at the top where you can stop sharing the screen. You go on the Zoom session first and then.
Yeah, sorry.
I can also.
Yeah, thank you.
Okay, so Jan, could you share your screen?
Yeah, but I'm not allowed to share it by...
1 second, yes. No, you should be.
Okay, so my contribution is a follow-up to work presented by my colleague Aday. Again, it's a site, it's a brownfield in western part of Bohemia, where more than 5,000 tons of HCH waste isomers were dumped in a dump of overburden of neighboring kaolin mine. So into the soil during the mining, into the waste soil, more than 5,000 tons of HCH were deposited, and damp leachate contaminated the surface water and sediment of the Ostrovsky stream and down gradient ponds. Here you can see the satellite photograph.
Sorry for interrupting you. If you want, you can also start the presentation mode. So now we see the whole presentation, but also the other slides.
Okay, sorry.
As you wish.
So here you can see the satellite photograph. In the center part, there is a dump. This is a former kaolin mine, and in the dump, the 5,000 tons of HCH isomers were deposited. And here the stream, Ostrovsky stream, flows and discharges into several ponds that are used for fishery. So it's very sensitive environment. The part located down gradient is also recognized as a Natura 2000 protection zone. This area will be solved or remediated by constructed wetland. But the first part of the project, we wanted to assess natural attenuation potential of the water, environment, and sediment of both the stream and the ponds. The methods comprised collection of surface water samples, stream and pond sediment, chemical analysis, and field measurements of surface water flow, and molecular biological analysis. The goal was to assess the potential for nature attenuation processes, namely absorption and biodegradation. Therefore, we measured concentration and we calculated the mass flux of contaminants and its reduction during the flow, surface water flow downstream. Here you can see the concentrations, the total HCH concentration in surface water that varies from first tenths of micrograms per liter to almost 100 micrograms. You can see the descending trend along the surface water flow. On the poster, I displayed also the vertical profile of concentration of HCH in sediment of the pond. You could see that at the inflow of the Ostrovsky stream, there are the highest concentrations of HCH, reaching up to 400 micrograms per kilogram. And here you can see the outcome of the molecular biological analysis performed with stream and surface water and sediment samples. We analyzed, or my colleague analyzed, both anaerobic biomarkers of degradation of HCH as well as aerobic biomarkers. And what is surprising that the surface water samples that are aerobic or oxic contains both anaerobic as well as aerobic biomarkers that indicate that both processes exist there. In sediment samples, especially in pond sediment, due to high content of organic matter, the environmentally strict anaerobic and anaerobic biomarkers only were identified. So to summarize the results, we found a quite significant reduction of mass flux of hexachlorocyclohexane during the flow of the surface water in approximately 1 kilometer. So during the residence time of approximately 4.5 hours, there's a reduction by 57%. And last year, we found the reduction of mass flux 94%. And based on the molecular analysis, in addition to sorption to stream sediments, aerobic biomarkers were exhibited high values in surface water samples. And anaerobic biomarkers were also detected, but with varying abundance along the course of the stream. With regard to sediment, anaerobic biomarkers, especially Dehalobacter and Dehalococcoides, dominate, indicating very good conditions for anaerobic biodegradation. And in Ostrovsky stream, where significant level of dissolved oxygen was found in surface water. So in sediment, both aerobic and anaerobic biomarkers were identified in high amounts. Thank you for your attention.
Thanks a lot to you. I will stop your screen sharing. I hope everybody was able to follow the presentation. There were some issues with the slides, but I think that was fine. We have a first question. I think it's addressed to Aday, and it's about were the beds planted with any vegetation?
The wetland bed which I presented, they didn't have any vegetation.
Okay, thanks. And second one that is also goes to you. You think that the biochar can be an identical performance for other types of pollutants, or even for a group of pollutants, because in some works results with and without biochar not show great differences. Thanks for the presentation.
Sorry. Okay. For other type of pollutants, right? You're asked.
Yes.
Yeah. It could work because the biochar is a strong sorbent, and in some researches, the biochar also enhanced the microbial biomass. So in my work, we're think that the biochar enhanced the microbial community to degrade our pesticides.
Okay, thanks a lot. And Anna Gorrito, if you have follow-up questions, I guess you can together open up a session in the coffee break and discuss that further. We also have a reminder by Carlos Arias that tomorrow on Thursday, there will be a workshop dedicated to the life what? I hope I said it right, at 3:30, but you can find it in the program anyways. And thank you all for presenting and for the nice questions. See you around.
Thank you. Bye-bye.
Thank you.